Hi, I'm Dr. Bobby. On behalf of a New Age Cosmetic White EdTech Healthcare Consultancy, I welcome all of you back to my daily lectures. We continue with the subject on genetics. Today we deal with recombinant DNA technology, which is also called gene cloning or molecular cloning, and is a general term that encompasses a number of experimental protocols leading to the transfer of genetic information, DNA. There is no single set of methods that can be used to meet this objective. However, a recombinant DNA experiment often has the following format. The DNA clone DNA, insert DNA, target DNA, or foreign DNA from a donor organism is extracted, enzymatically cleaved, cut, or digested, and joined, ligated to another DNA entity, a cloning vector to form a new recombined DNA molecule, cloning vector, insert DNA construct or DNA construct. This cloning vector insert DNA construct is transferred into and maintained within a host cell. The introduction of DNA into a bacterial host cell is called transformation. Those host cells that take up the DNA construct, transform cells, are identified and selected, separated or isolated from those that do not. If required, a DNA construct can be created so that the protein product encoded by the cloned DNA sequence is produced in the host cell. Recombinant DNA technology was developed from discoveries in molecular biology, nucleic acid enzymology, and the molecular genetics of both bacterial viruses, bacteriophages, and bacterial extrachromosomal DNA elements, plasmids. However, recombinant DNA technology would not exist without the availability of enzymes that recognize specific double-stranded DNA sequences and cleave the DNA in both the strands of these sequences. Restriction enzymes or restriction endonucleases. Nucleases that cut nucleic acid molecules internally are endonucleases, and those that degrade from the ends of nucleic acids are exonucleases. And as we proceed further, I'd like to extend our thanks and courtesy to the authors of this book, Molecular Biotechnology, by Glick, Bernard, Jack, and Cheryl. Thank you so much for these valuable notes. Many students would benefit from these, and I request that all candidates listening to my lectures to please make notes, but kindly do not commercialize or sell any portion of these notes or lectures. Please use them for your personal benefit of studies and passing your exams. Thank you. This is continuing to this subject proper. This is a schematic. And this diagram shows the recombinant DNA cloning procedure. DNA from a source organism is cleaved with a restriction endonuclease and inserted into a cloning vector. The cloning vector insert target DNA construct is introduced into a host cell, and those cells that carry the construct are identified and grown. If required, the cloned gene can be expressed and transcribed and translated in the host cell, and the recombinant protein can be harvested. So this is the picture of that. Take a screenshot for your reference and make notes, right? And the next topic will be restriction endonucleases. For molecular cloning, both the source DNA that contains the target sequence and the cloning vector must be consistently cut into discrete and reproducible fragments. It is only after bacterial enzymes that cut DNA molecules internally at specific base pair sequences were discovered that molecular cloning became feasible. These enzymes are formally designated by type 2 restriction endonucleases, despite the fact that there are other kinds of restriction endonucleases, type 1, type 3, and type 4. And type 2 restriction endonucleases are commonly called restriction endonucleases or simply restriction enzymes. One of the first type 2 restriction endonucleases to be characterized was from the bacterium Escherichia coli and was originally designated E. KORAH. More recently, it has been proposed that the use of italics for naming and restriction endonucleases can be abandoned. Here we have implemented this recommendation. EcoRI is a homodimeric protein. It is made up of two identical proteins that binds to a DNA region with a specific palindromic sequence, recognition site, or binding site. In other words, the sequences of nucleotides in the two strands of the binding site identical, with either is read in the same polarity, that is 5' to 3'. The eco-recognition sequence consists of 6 base pairs BP and is cut between the guanine and adenine residues on each strand. EcoRI specifically cleaves the internucleotide bond between the oxygen of the 3' carbon of the sugar of one nucleotide and the phosphate group attached to the 5' carbon of the sugar of the adjacent nucleotide. The symmetrical staggered cleavage of DNA by EcoRI produces two single-strand complementary cut ends, each with extensions of 4 nucleotides known as sticky ends. The diagram shows a symmetrical staggered cleavage of a short fragment of DNA by the type 2 restriction endonuclease EcoRI. The large arrows show the sites of cleavage in the DNA backbone. S-deoxyribose sugar, P-phosphate group, OX hydrox group. The EcoRI recognition sequence is highlighted by the dashed line. Please make a note of the diagram here, which is the illustration here. It could help you in your notes. Each single-stranded extension terminates with the 5' phosphate group and the 3' hydroxyl group of the opposite strand is recessed. In addition to EcoRI, more than 3,700 type 2 restriction endonucleases with about 250 different recognition sites have been isolated from various bacteria. The naming protocol for these enzymes is the same as that for EcoRI. The genus is the capitalized letter, and the first two letters of these species names are in lowercase letters. The strain designation is occasionally added to the name, such as R in EcoRI, or the serotype or the source bacterium is sometimes noted, such as D in Hind 3. Roman numerals are used to designate the order of characterizations of different restriction endonucleases for the same organisms. For example, HPA1 and HPA2 are the first and second type 2 restriction endonucleases that were isolated from Haemophilus influenzae. Palindromic sequences where most type 2 restriction endonucleases bind and cut a DNA molecule are within the recognition sites. Some restriction endonucleases digest cleave DNA, leaving 5' phosphate extensions, protruding ends or sticky ends with recessed 3' hydroxyl ends. Some leave 3' hydroxyl extensions with recessed 5' phosphate ends. And some cut the backbones of both strands within a de-recognition site to produce blunt-ended, flush-ended DNA molecules. The lengths of the recognition sites for different enzymes can be 4, 5, 6, 8 or more nucleotide pairs. Because of the frequency with which the recognition sites occur in DNA, restriction endonucleases that cleave within sites of 4 cutters and 6 cutters nucleotide base pairs nucleotide pairs are used for most of the common molecular cloning protocols. The importance of the type 2 restriction endonucleases for gene cloning cannot be overstated. Blunt-end cleavage of a short fragment of DNA by the type 2 restriction endonuclease Hind 2. The large arrows show the sites of cleavage in the DNA backbone for the abbreviations, as shown in the legend 3.2. The Hind 2 recognition sequence is highlighted. Take a screenshot and make your notes, please. This is a table that shows the recognition sequences of some restriction endonucleases. I'll just name the enzymes. The rest you can just make notes after you've taken screenshots. The enzymes will be EcoRI, BAM1 and 2, PST1, SHO3AI, PBU2, HPA1, HAE3, and NOT-I. Arrows denote cleavage sites. Okay, going to the subject. When a DNA sample is treated with one of these enzymes, the same set of fragments is always produced, assuming that all the recognition sites are cleaved. In addition, Ready access to a variety of restriction endonucleases adds versatility to gene cloning strategies. Type 2S restriction endonucleases form a subgroup of the type 2 category of restriction enzymes and are occasionally used for cloning and other molecular studies such as multiplex polymerase sequencing that I discussed in chapter 4. It's coming up soon. These enzymes have the fascinating feature of cutting DNA, usually in both strands, a fixed number of nucleotides away from one end of the recognition site. Moreover, any particular sequence of nucleotides may be present between the binding sequence and the cut sites. The cleavages for most type 2S restrictions enzymes are staggered. For example, the FOC1 restriction endonuclease binds to the sequence shown here and cuts 9 nucleotides downstream on the upper strand and 30 nucleotides downstream on the lower strand, producing a recessed 3' hydroxyl end and a 4 nucleotide extension at the 5' phosphate end. One representation of the recognition sequence and cut sites of the FOC1 restriction endonuclease is where N denotes A, C, G, or T. Of course, with the single letter code, it is understood that the nucleotides N opposite each other are base paired. A simpler notation is the sequence shown here, and perhaps the simplest is as shown here. This one. Examples of some type 2 restriction endonucleases are shown in table 3. We'll go to that. It should be noted that a few type 2 restriction and endonucleases cleave DNA both upstream and downstream from the recognition sites. Under natural conditions, bacteria use restriction endonucleases to cleave foreign DNA, such as that of infecting bacterial viruses, bacteriophages, and have developed systems that protect their own DNA from being upgraded. Most often, methylation of the cytosine residues of a restriction endonuclease site in the host DNA prevents restriction endonucleases from cutting at these sites, but the non-methylated sites of foreign DNA are vulnerable to attack. With the characterization of large numbers of restriction endonucleases from various bacteria, interesting relationships have been observed. In some instances, different phosphodiester bonds. The table 3.2 shows the restriction endonucleases: AQ1, BFU1, A1, BSMB1, ECI1, FOC1, HGA1, MLY1, and MME1. You can take notes on these. Continuing with subject, and within the same recognition site are cleaved by restriction endonucleases from different organisms. For example, the restriction enzyme XMAI and SMAI both recognize the sequence, but XMAI cleaves after the first 5' cytosine in each strand and produces 5' phosphate extensions, whereas SMAI generates blunt ends by cleaving between the shown base pair, the CGGC base pair in the middle of the recognition site. The palindromic sequences where most type 2 restrictions endonucleases bind and cut a DNA molecule are within the recognition sites. Some restriction endonucleases digest cleave DNA, leaving 5' phosphate extensions, protruding ends, or sticky ends with recessed 3' hydroxyl ends, and leave 3' hydroxy extensions with recessed 5' phosphate ends, and some cut the backbones of both strands within a recognition site to produce blunt-ended, flush-ended DNA molecules. The lengths of the recognition sites for different enzymes can be 4, 5, 6, 8, or more nucleotide pairs. Because of the frequency with the recognition sites occur in DNA, restriction endonucleases that cleave within sites of 4 cutters and 6 cutters nucleotide pairs are used for most of the common molecular cloning protocols. The importance. This lecture continues in the next file. So, the importance of the type of restriction endonucleases for gene cloning cannot be overstated. This blunt-end cleavage of a short fragment of DNA by the type 2 restriction endonuclease hind 2, the large arrows show the sites of cleavage in the DNA backbone. For abbreviation, see the legend to figure 3.2. The hind 2 recognition sequence is highlighted. Each single-stranded extension terminates with a 5' phosphate group and 3' hydroxyl group of the opposite strand is recessed. In addition to KORAH, more than 3,700 type 2 restriction endonucleases with about 250 different recognition sites have been isolated from various bacteria. The naming protocol for these enzymes is the same as that for KORAH. The genus is the capitalized letter, and the first two letters of the species named are in lowercase letters. The strain designation is occasionally added to the name, such as R in KORAH, or the serotype of the source bacterium is sometimes noted, such as D in Hind 3. The Roman numerals are used to designate the order of characterization of different restriction endonucleases from the same organisms. For example, HPA-I and HPA-2 are the first and second type 2 restriction endonucleases that were isolated from Haemophilus parainfluenzae. The palindromic sequences where most type 2 restriction endonucleases bind and cut a DNA molecule are within the recognition sites. Some restriction endonucleases digest cleaved DNA, live in 5' phosphate extensions, protruding ends, or sticky ends with recessed 3' hydroxy ends. Some leave 3' hydroxyl extensions with recessed 5' phosphate ends, and some cut the backbones of both strands within a recognition site to produce blunt-ended, flush-ended DNA molecules. Figure 3.3. The lengths of the recognition sites for different enzymes can be 4, 5, 6, 8, or more nucleotide pairs. Table 3.1. Because of the frequency with which their recognition sites occur in the DNA, restriction endonucleases that cleave within sites of 4 cutters and 6 cutters nucleotide pairs are used for most of the common molecular cloning protocols. The importance of the type 2 restriction endonucleases for gene cloning cannot be overstated. Blunt and cleavage of a short fragment of DNA by the type 2 restriction endonuclease, end 2. The large arrows show the sites of cleavage in the DNA backbone. For abbreviation, see the legend 2, figure 3.2. The Hind 2 recognition sequence is highlighted. This is table 3.1. It shows the recognition sequences of some restriction endonucleases. KORAH, BAM-HI, PST1, SAU3A1, PBU2, HPA1, HEAT3, and NOT1. So this has been dealt with already. So I'm just showing you for the tables. The restriction enzymes XMAI and SMAI both recognize the sequence, but XMAI cleaves after the first 5 cytosine in each strand and produces 5' phosphate extensions, where SMAI generates blunt ends by cleaving between the base pair, the middle of the recognition site. The first restriction endonuclease that is discovered to bind to a particular recognition site is designated the prototype. Any individual restriction endonuclease that attack the same sequence as the prototype are called isoschizomers. For example, the restriction endonucleases XO and PAER71 from different organisms both have the same recognition sequences and cleavage locations. Isoschizomers that cleave at different positions within the same recognition site and neoschizomers. On the other hand, restriction endonucleases that produce the same nucleotide extensions but have different recognition sites are designated isochordamers, example BAM-HI and SAU3AI. In some cases, a restriction endonuclease will cleave a sequence only if the cytosines of the recognition site are not methylated, whereas another restriction endonuclease will cut the same sequence if these cytosines are methylated. For example, HPA2 cleaves only non-methylated sites. An MSP1, an isoschizomer of HPA2, cuts the sequence regardless of cytosine methylation. This pair of restriction endonucleases is often used to determine the methylation status of genomic DNA. If a DNA molecule is not cut by HPA2 but is cut by MSP1, then the recognition site is methylated. If both restriction endonucleases cleave a DNA molecule, then the site is not methylated. Physical maps that designate the relative positions of restriction endonuclease sites on a specific piece of DNA can be reconstructed by treating the DNA molecule singly with different restriction endonucleases, and then with combinations of the same restriction endonucleases. The positions of the cleavage site can be deduced from an analysis of fragment sizes, which are determined by agarose gel electrophoresis. By way of illustration, the fragment sizes produced by various digestions are shown. You could have a look at the figure 3.4 of the neoschizomers. 4 restriction endonucleases bind to the same recognition site and cleave at different positions. The restriction endonucleases and cleave sites arrows are color coded. Cas1 red, now 1, 2. Cas1 is red, now 1 is 2 blue. SFI is black, BBEI is green. A number of other restriction endonucleases such as NDA1, etc. that bind to and cleave the sequence are not shown. In figure 3.5a, it can be deduced that the linear piece of DNA has 2 BAM-HI sites and 2 EcoRI sites in a different order with a specified number of base pairs separating the sites. More specifically, the sizes of fragments produced in each single digestion can be compared with those from double digestion to determine the position of the restriction endonuclease sites and to generate restriction endonuclease site map. Restriction endonuclease map. In the example shown in figure 3.5, the analysis goes as follows. Because each single digestion produces 3 fragments from a linear DNA molecule, the original piece of DNA must contain 2 sites for each of the restriction endonucleases, the 3000 BP fragment. We'll come to this special note later. We'll read this before proceeding to subject matter proper. Box 3.1. Gel electrophoresis. Courtesy to the authors for such invaluable notes. Gel electrophoresis is a commonly used technique for resolving proteins or nucleic acids. In general, a sample of one particular type of macromolecule, protein, DNA, or RNA, is placed in a well at or near the end of a gel matrix gel. The composition of electrophoresis gel is a semi-solid, open meshwork of interlinked linear strands. A gel is cast as a thin slab with a number of sample wells. After the wells of a gel are loaded with sample, an electric field is applied across the gel and charged macromolecules of the same size are driven together in the direction of the anode through the gel as discrete invisible bands of material. The distance that a band moves into a gel depends on the mass of its macromolecules and the size of the openings, both sides of the gel. The smaller macromolecules travel further than the larger ones. The progress of gel electrophoresis is monitored by observing the migration of a visible dye, tracking dye, through the gel. The tracking dye is a charged low molecular weight compound that is loaded into each sample well at the start of a run. When the tracking dye reaches the end of the gel, this run is terminated. The bands which are aligned in a lane under each well are visualized by staining the gel with a dye that is specific for protein, DNA, or RNA. Discrete bands are observed when there is enough material present in a band to bind the dye to make the band visible, and when the individual macromolecules of a sample have distinctly different sizes. Otherwise, a band is not detected. If there is a little or no difference among the sizes of the macromolecules in a concentration sample, a smear of stained material is observed. The intensity of a stained band reflects the frequency of occurrence of macromolecule in a sample. The molecular mass, molecular weight of a gel fractionated macromolecule band is determined from a standard curve that is based on a set of macromolecules of known molecular mass size markers that covers the separation range of the gel system and is run in one or both of the outside lanes, calibrated lanes of the same gel as the samples. The logarithm of the molecular mass of a size marker is related to its relative mobility, RF, through a gel. The value of RF is defined as the distance traveled by a band divided by the distance traveled by the tracking dye ion friend. The relationship between the logarithm of the molecular mass of each size marker and its R value is plotted. Then with the standard curve, a molecular mass is calculated for each band in a lane. The units of molecular mass for proteins and double-stranded and single-stranded nucleic acids are daltons, base pairs, and bases, respectively. The size markers are included in the same gel as the samples because the extent of mobility of a macromolecule varies from one electrophoretic run to the next. Polyacrylamide is the preferred gel system for the separating proteins. Copolymerization of monomeric acrylamide and the cross-linker bisacrylamide forms a lattice of cross-linked linear polyacrylamide strands. The pore size of a polyacrylamide gel is determined by the concentration of acrylamide and the ratio of acrylamide to bisacrylamide. For many applications, a protein sample is treated with the anionic detergent sodium dodecyl sulfates SDS before electrophoresis. The SDS binds to proteins and dissociates most multi-chain proteins. Each SDS-coated protein chain has a similar charge-to-mass ratio. Consequently, during electrophoresis, the separation of the SDS protein changes base primarily in size, and the effect of conformation is eliminated. SDS polyacrylamide gel electrophoresis with a 10% polyacrylamide gel results proteins that range from 20 to 200 kilodaltons kDa. Agarose, which is a polysaccharide from seaweed, is used routinely as a gel matrix for the electrophoretic separation of medium-sized nucleic acid molecules. A 1% agarose gel can resolve duplex DNA chains that range from 600 to 10,000 BP base pairs. Specialized agarose gel electrophoresis systems are available for fractionating DNA molecules with millions of base pairs, denatured DNA, and denatured RNA. In addition, for specific purposes, polyacrylamide gels are used for separating DNA molecules. For example, DNA chains that are as small as 6 bases and that differ from each other by one nucleotide can be resolved with a 20% polyacrylamide gel. Shown above is a mapping of restriction endonuclease sites. A is a restriction endonuclease digestion and electrophoretic separation of fragments, as you see here. This one. And a purified linear piece of DNA is cut off with the KORAH and BAM-HI separately, single digestions, and then with both enzymes together, double digestion. The horizontal lines under digestion conditions represent schematically the location of the DNA fragments, bands, and the lanes of the gel after electrophoresis and staining of the DNA with ethidium bromide. The numbers denote the lengths of the digestion products, fragments, in the base pairs. B. Restriction endonuclease map derived from the digestions and electrophoretic separation shown in panel A. And so is produced by the single eco-RI digestion remains intact after the double digestion, whereas the 8500 to 5000 BP eco-RI fragments are cleaved. Therefore, the two eco-RI sites are 3000 base pairs apart with no intervening BAM-HI site, and there is a single BAM-HI site within each of the 8500 and 5000 BP eco-RI fragments. The 9500 BP fragment that is produced by the BAM-HI digestion is cleaved by eco-RI and the double digestion into 3 pieces. 2500 plus 3000 plus 4000 will be 9500 base pairs. Therefore, the 2B BAM-HI sites lie 2500 and 4000 base pairs to either side of the eco-RI sites. Digestion with BAM-HI cleaves the 8500 BP eco-RI fragments and 2500 and 1600 BP fragments, and one of the eco-RI sites is 2500 BP BAM-HI site. So the 1600 BP region must include one of the ends, the resin molecule. Use the same logic. We also note that digestion with BAM-HI cuts the 1500 BP eco-RI fragment into 1000 and 4000 BP fragments, and that one of the eco-RI sites is 40,000 BP from a BAM-HI site. Therefore, the 1000 BP is the region The base pair region must include the other end of the original molecule in the final map, figure 3.5, be the assigned location of the restriction endonucleoside are consistent with the fragment lengths that were observed in each of the digestion reactions.